Have you ever wondered why certain characteristics are more common in some groups than others, or how life forms manage to adapt to new challenges over countless generations? It is a question that speaks to the very fabric of our biological heritage and the dynamic nature of evolution itself. Population genetics research fundamentally aims to understand the distribution and change of genetic variation within and between populations. It is the scientific field that deciphers the genetic blueprint of groups, not just individuals, to reveal their evolutionary stories. This understanding is critical for grasping how populations persist, evolve, and sometimes even disappear. The why behind this exploration is deeply rooted in the mechanisms of evolution. At its core, evolution is the change in allele frequencies in a population's gene pool over time. Population genetics provides the quantitative tools to measure these frequencies and identify the forces driving their shifts. There are four primary evolutionary forces at play. First, natural selection favors traits that enhance survival and reproduction. Second, genetic drift causes random fluctuations in allele frequencies, especially pronounced in smaller populations. Third, gene flow involves the movement of genes between populations through migration, which can introduce new variations or homogenize existing ones. And finally, mutation continually introduces novel genetic variants, providing the raw material for all other evolutionary changes. By meticulously analyzing how these forces interact, researchers can reconstruct the historical movements of populations, identify genes under selection, and predict future evolutionary trajectories. This understanding translates into powerful applications across various fields. For instance, in conservation biology, it helps identify endangered populations with dangerously low genetic diversity, guiding efforts to maintain their long-term viability. In medicine, population genetics can shed light on why certain diseases are more prevalent in particular ethnic groups or how populations might respond differently to medications, leading to more personalized healthcare approaches. It also plays a vital role in tracing human ancestry, agriculture for crop improvement, and even forensic science. Modern tools like high-throughput DNA sequencing and sophisticated statistical modeling allow scientists to process vast amounts of genetic data, revealing patterns that were once invisible. A foundational concept in this field is the Hardy-Weinberg equilibrium, which describes an idealized population where allele and genotype frequencies remain constant across generations in the absence of evolutionary influences. Real populations rarely meet these conditions, but the deviation from Hardy-Weinberg equilibrium provides a powerful null hypothesis for detecting evolutionary forces at work. Understanding demographic history, such as past population bottlenecks or expansion events, is also crucial as these historical occurrences leave distinct genetic signatures that population geneticists can interpret. This allows us to trace the origins and dispersal of species, including our own. Ultimately, the purpose of population genetics research is to provide a comprehensive framework for understanding biological diversity and adaptation. It offers a powerful lens to view the past, present, and future of life on Earth, informing critical decisions in areas like public health, environmental conservation, and our fundamental quest to comprehend human origins and diversity.